The next speaker is Professor Charles Wondji from the Liverpool School of Tropical Medicine, who is a vector biologist and professor and heads the Center for Research in Infectious Diseases that's found in Yaoundé, Cameroon. Professor Wondji.
Yes, my talk will focus on vector control and the challenge from the emergence of resistance to insecticide. Okay, vector control, as we know, remain a cornerstone of malaria transmission, malaria prevention, sorry, and the recent decline in malaria burdens from the year 2000 has been mainly contributed by vector control, as it has been shown clearly by a study published in Nature by Bat et al. 2015, that long-lasting insecticide nets and indoor residual spraying have significantly contributed to this reduction at the level of 81%, and averting around more than 600 million clinical cases of malaria. And this success has coincided with the scale-up of bed nets across the continent. But as previously mentioned by Abdullah, there has been a rebound in malaria cases, and this has been shown to coincide also with the growing report of insecticide resistance in major malaria vectors. And as you can see on the slide there, the red dots there indicate the presence of insecticide resistance in the major malaria vectors in Africa. And that could be associated with the spread, with the increase in malaria cases that the WHO reported last year. And we also need to bear in mind that there are just limited number of insecticides available for public health control. And this also could be the reason of the development of resistance, because there is what we call an over-reliance on a limited number of insecticides families, the main one being the pyrethroids. As you can see there on that graph, pyrethroids are the main insecticide class used for malaria control. Among the four, we have the organochlorine, organophosphate, carbamate, pyrethroids. And why are we using mainly pyrethroids? The reason is simple. It's because these are the less toxic to humans. So they are the main one at the moment recommended for bed net impregnation. And should we worry about insecticide resistance, or is it just an academic issue? And there are more and more evidences that this is serious, as shown by this paper published last year in Lancet, showing that when bed nets are compared, one where you only have pyrethroid and the other one having what you call PBO is a synergist, which can block the action of cytochrome P450s, which confer resistance. It has been shown that when the PBO nets are used, there's significantly lower prevalence of parasitemia, lower prevalence of severe or to moderate anemia, and lower entomological inoculation rate, showing overall that because of resistance, bed nets are losing their efficacy. And this is a strong evidence that even led WHO to also issue an interim endorsement for the PBO-based nets. And when there is resistance, what should we do? I put this slide just to summarize what are the potential strategies available for resistance management. There are mainly three, what we call first mixtures, where we can apply two mixed compounds so that individual mosquitoes are exposed at the same time to each compound. And the second strategy is what we call rotation, where there's a temporal alternation of unrelated insecticides, as you can see here from year 1, 2, and 3. And the third one is what we call mosaic, where we apply insecticide in adjacent areas simultaneously with different insecticides, of course. But for these strategies to be implemented, it's so critical for us to understand how mosquitoes are acquiring resistance. And therefore, we need to understand what are the mechanisms in place. And overall, there are four main mechanisms. The first one, or I can say the biochemical ones. There are two. We have what we call the target site resistance, where there will be a modification of the target site in the mosquitoes, preventing insecticides to bind to the target, and that mosquito will become resistant. We also have the metabolic resistance. Mosquitoes are able to overexpress certain detoxification enzymes, which will degrade the insecticides before it can reach the target. We also have what we call behavioral resistance. Mosquitoes are able to change their behavior. They can bite earlier to avoid the period when people are under the bed nets, for example. But there is also what we call reduced penetration, where, for example, mosquitoes are able to thicken the cuticle of the body and then limiting access of insecticides in the mosquitoes. And another reason why it's important to understand the mechanism of resistance is because to implement an efficient resistance management strategies is so important that resistance is detected at a very early stage, because this is when it's easier to manage the resistance. And it's only when we have molecular tools, molecular assays, that we are able to detect resistance when when the allele is still at the heterozygote state. When we wait, when you have homozygote resistance, and it's only at this stage you can use simple bioassay, but usually it's a bit late to manage the resistance. So it's critical that we get the tools, the molecular tools, to detect resistance early. And for the target site resistance, KGR for example, we have been successful in doing this because the molecular assay for these mechanisms was designed more than 20 years ago. And as you can see, we can detect when a mosquito is susceptible, homozygote resistant, and heterozygote. And this has been really helpful in detecting the spray of resistance. As you can see here, those black dots represent the presence of the resistant allele, and it's now widespread across Africa for this target site resistance. But for the metabolic resistance mechanisms, it's a bit more challenging, and the progress has been slow. And why has it been slow? Mainly because there are many enzyme families involved, from the P450, GSTs, esterases, and others. But even so, we have more than 200 enzymes which potentially can confer resistance, and the challenge is which one is involved. And there are also many other mechanisms that could contribute to the resistance. And what I could say is that recently important progress has been made, and I will now show you what those progress are. We have recently been able for the first time to detect the causative markers of what we call the P450 resistance. And because we have done so, we designed a simple PCR RFLP assay, which is now helping us to detect resistance easily. And these markers has a strong correlation with the phenotype, which is an important step forward. And having done that, we have been also successful in doing the same for another gene. So showing that when we are able to learn from recent success to make further progress in this direction. This is also a P450 gene. And that was for one species, but because, as you know, there are many species of malaria vectors, and the progress is being done in other species as well. This is Anopheles gambiae, where the work done and published 2 years ago have identified key loci contributing to resistance. And using the whole genome sequencing, that was the case. We have also been able to show that actually it is the bednet scallop which is selecting for the resistant allele. As you can see, this is before bednet. There was no reduced diversity in Phaenostis, but after bednet, the resistance has been selected so much because of bednet scallop. But the challenge for those working in this field is that the resistance mechanism could be really complex. This is another type of mechanisms that was recently published, that was 3 months ago, by a team at the Liverpool School, showing that mosquitoes not only they overexpress specific genes, but they can increase the number of copy of these genes to further break down insecticides. Another challenge is that that was last year, there are novel mechanisms also being reported, showing that mosquitoes are actually evolving complex mechanisms to develop resistance to the current insecticides. Someone may ask, why do we bother identifying these mechanisms? One of the benefits is, first, when we have the tools, we can easily detect resistance and monitor how this resistance is spreading across the continent. For the 2P450 genes I just told you, we were able to show that it's highly present in Southern Africa, but so far it is absent in other regions. And we also noticed that another markers that we had detected before is only present in West, Central, and East, not in Southern. And so just by doing this work, it could show that there seems to be a barrier to gene flow between African regions. So the mosquitoes are not developing resistance in the same way. So this is another challenge. And even for the new methods like gene drive, if you just release mosquito in one part, it doesn't guarantee that the gene will spread across the continent. So we need to bear that in mind. Second advantage is that when we now have these tools, we can assess how resistance, particularly metabolic resistance, is impacting the efficacy of bed nets. This is a work that we have done in Cameroon using experimental hot trial. We have shown that the bed nets with the PBO are more efficient. As you can see, the mortality with PBO net is very high compared to pyrethroid only. But the question we ask is, those mosquitoes surviving exposure to bed nets, what is the genotype? What we establish is that those alive after bed net exposure were either homozygote resistant or heterozygote, whereas those dead were mainly susceptible. And furthermore, the impact on malaria transmission is also seen because when a mosquito is resistant, it is more likely to blood feed. So showing that resistance can increase malaria transmission. And we have also been able to ask the question of what happens when a mosquito is resistant to multiple genes. We have shown that in that case, it's able to survive even better. This is double homozygote resistant against bed nets in the experimental huts. None of the double homozygotes died. But in contrast, all the double homozygote susceptible nearly died. So it just shows that with these tools, we can understand the impact of resistance much better. Another factor is what we call fitness cost of resistance, because to manage resistance, like rotation, you need to ensure that there is a fitness cost. And because we now have the markers, we are able to look how is resistance impacting the fitness of mosquitoes. We show, looking at the larva stage, that when mosquitoes are resistant, they are less likely to survive from L1 to L4 stage. And this is a good news. It means that if we implement resistance management strategy early, we can actually manage resistance because those resistant are less likely to be fit as the susceptible. And that was confirmed when we look at the adults. We look at in the absence of insecticides, we noticed that those from over 10 generations, the frequency of susceptible is increasing, as you can see there, again, confirming that if we implement management strategy on time, we could manage resistance. Another element that we notice is that because of resistance, there is clearly an increase in malaria transmission. This is what we show with one markers in GST E2. Those homozygote resistant were more infected to Plasmodium. And then, as we close, is it all bad news? I will say no, because in terms of insecticide-based interventions, there are new products being made available. You have new insecticides, which have different mode of actions than the current pyrethroids, and these have been recommended by WHO. So it means that we have more tools. But also, even for bed nets, there are new bed nets being released, those based on dual active ingredient, or more and more available, and there is the consortium by IVCC releasing soon new insecticides, which should make it easy to control mosquitoes in the future. And there are also alternative control, like gene drive, which is in the pipeline, and we have heard about that, which hope to reduce malaria transmission as well. So, as we close, what's the way forward? I think we need to do all we can to ensure we maintain the effectiveness of current tools. We need also to better manage resistance using genomic approach. We need also to look at alternative methods like gene drive to better control vector control of mosquitoes and help reduce malaria transmission. I would just like to close by acknowledging the team behind all this work. Thank you for your attention.